Hi, everybody. This is the next meeting now. Good to see you all, Mark, Zarina, Jim. Good to see you, Richard. And we do have slides this time, so I'm going to paste the link. I have to find them. Let's wait a few moments.
Hey, are you okay with me taking a screenshot now with everybody on the screen to show how we work?
All right, welcome everybody. This is the second meeting after the NIH HCA meeting, and Jim Gee and myself, and I think also Peter Hunter is trying to join using low internet. We are very glad that you're all back. Good to see you again. I'm going to share my slides here to have everybody on the same page. And again, this is the anatomical structures and cell types meeting. And we have an entire hour. And those of you which haven't been able to to join us for the first meeting, we actually had a number of experts volunteer to create these anatomical structures, cell types tables for their favorite organ. And so those of you which haven't seen that growing table, during the NIH HCA meeting, we created a table of different organs, here rows, that different teams care about. And we also tried to identify which efforts funded by NIH and otherwise care about which of these organs. And you also see a total on the right-hand side. And then in the last meeting, we identified a number of organs for which we want to try to get to those tables. And just in case you haven't seen one of those tables, here's the original table by Sanjay, and he's going to present it in a moment. But basically, you have structural regions, you have substructures, you have cell types, you have abbreviations, you have subsets of marker genes, and then you also have a space for comments. And so in the last meeting, we discussed the tables that were created for kidney and spleen within HubMap. But then we also looked at how we can connect to other efforts that are also working on these tables. And so we have still the introductory slides, which were used at the NIH HCA session, linked from here. So you all should have that link to the slides now. If not, maybe Ellen could paste that link one more time into the chat log for those which joined late. We also have the recording from the 4.9 call available via this link here. Today's topic is about looking at new tables that have been created in the last 4 weeks. And we had a number of experts which volunteered to create these tables, and we had others which volunteered to review and provide expert feedback. We will today review this initial set of tables. If you happen to be one of the authors of those tables or a reviewer, please make sure to add your information to this Google Doc, which Ellen kindly provided, so that we can give credits to all of those which really contributed to this effort. We will then also discuss the editorial process to curate those tables. And this is a first draft, and so we would welcome your feedback on it. I think it's a very unusual type of scholarly output. It's not data, it's not code, it's not a publication, it's a table structure that will evolve over time. And then we also have a number of ontology experts joining today. And we will discuss how to best link those tables to existing ontologies, for instance, Uberon. And then if there is time, and I hope there is, we will discuss how to link these tables also to the 3D reference organs, such as visible human organs in HubMap or scaffolds used in Spark or Ellen's Atlas 3D, which came out just recently. So everybody should have access to those slides. I hope everybody can put their name down here so that we have a good understanding of who is in this call. We could also, in the middle, we might just take a screenshot also of everybody who is in there so that we have some understanding of who is interested in this effort and can make sure that you all get an invite to the next meeting.
Is this the right link?
Yes, hi Sanjay.
Sorry, yeah, I was clicking on the wrong link, thank you, yeah.
So please add your names in here so that we also know who is available from which effort. And I think Jimgi, you might like to add yourself, and I'm going to add myself. So I guess this is all editable by all of us.
Kati, could you please put the link for this in the chat?
Is the Google Docs, right? The first one?
Yeah, so I think if you join a little late, then you wouldn't see it anymore. So I'm just gonna paste it one more
I can take care of putting that stuff back in.
Thank you, Ellen. And Lisa, I think, also posted it one more time. Excellent. All right. Sanjay just joined. Welcome, Sanjay.
Some issues with your sound, or am I having issues? Is it only me?
I think somebody is typing. Maybe Ellen, mute yourself.
No. The sound is variable. So if it's not me, that's okay.
Okay, I will get closer. So Sanjay, if you want to present the first table that got revised over the last weeks, that would be wonderful.
Yeah, so if you want to go, I think to your next slide, you have it already, right? I'm not sure if you folks can see it, but what we have been doing in KPMP is the group is based on the data coming from KPMP on single cell or single nucleus or regional transcriptomics is identifying cell types and the genes which are most specifically related to those cell types, and then trying to go higher order to identify the subregions that the community is using, and then the higher order anatomy of that is the eventual goal. So this table has been generated, with the that we have been doing in KPMP, and then we are now interfacing with the HubMap folks, the Vanderbilt group from Jeff Spraggins, and then the next task is to work with ontologists. So the KPMP has a task force. There was a meeting in January which was attended by ontologists and members outside the community to come up with consensus definitions or consistent definitions so everybody can use. And there has been, some people, definitions people can agree, and some are in use that sort of are not consistent with what's in the literature or in the ontology databases and so forth. So we are now, obviously trying to get feedback to see how to resolve those inconsistencies. And I think Kati forwarded me, some of the work that was done by Chris Mungle, and which, there were, some inconsistency in ontology databases regarding some of the terms, like for in the definitions of renal corpuscles and what should be in it, what should be in cell types in the glomerulus versus in the capillary. And I think this is where we are at, and we will look forward to discussing these and resolving this, bringing it back to the groups of pathologists, nephrologists, and the biologists, because some of the terms are quite complicated, and over the years, the meanings are different to different people. So I think that's where we are at with this.
Thank you so very much, Sanjay. And I see Chris and Melissa here on the call. And Chris, if you want to guide us through this graph on the left-hand side and explain a little bit more what you did here. Thanks so much for doing this and on a very short timeline.
Yes, thank you. Yeah.
Sure, yeah. So what I did was I just took at random the first kind of block of this table where we looked at the renal corpuscle, and I mapped all of the terms within there to either Uberon or CL. I made a few judgment calls, like, a mesangial cell. According to cell ontology, there's both glom mesangial cell and an extra glom mesangial cell. So I think I just kind of arbitrarily chose one of those as the mapping. You don't see the full table on the right here, but I think you're familiar with it. And then after I did that, I visualized the ISA and Partov relationships in CL and Uberon. So remember, CL is the cell type ontology, Uberon is gross anatomy, and there's Partov links between the two. So what you can see here is the Uberon terms in yellow, the CL cell type terms in white, and the ones with the thick borders around them are the ones from this ASCT table. So there's differences in nomenclature. You know, as I said, the mesangial cell I mapped to the GLOM one. Podocyte, you know, I took to be a synonym for GLOM visceral epithelial cell. And this is based on the synonym designation within the cell ontology. We actually, in all of our ontologies, we have alternate labels, and we annotate those alternate labels with, you know, with usage, whether it's an exact or an imprecise synonym. And we aim to also annotate those synonyms with, you know, publications where that synonym may have been used and so on. So there's terms, the terms that are not bolded are ones that are not in the table. So these are maybe, you know, if I had more time, I could have like maybe grayed those out or something and showed the direct inferred links between the ASCT terms. But what this essentially shows is that the two are largely consistent. You've got renal corpuscle right there at the top, and then you've got the next level division is between the GLOM and the GLOM capsule, which we're calling the Bowman's capsule, which I'm taking to be exact synonyms. But there were a couple of differences. So we, in the cell ontology, had the podocyte being within within Bowman's capsule, and whereas over here you have it as being within the GLOM. And so I actually opened a ticket for this in the Uberon tracker. And just as a nice example of community usage of these ontology, there's already, you know, a couple of comments from other people about, you know, the difficulties with podocyte and what does it actually mean and where should it actually be placed and so on. So we could definitely dive into the anatomy here, but I think this is just, it was just a really nice exercise in terms of how do you map these terms. You know, some of them may be imprecise, there may be differences in the community about how these things are defined. And that's without even getting into the genes that are markers for the cell types. And I can paste a link for the issue in the tracker, and we've got comments from a couple of people, and including Anne-Marie Massey, who's I think is part of this.
Yes, Anna Maria Massey.
Hi there. Yeah, thanks for your comments.
Yeah.
Thank you.
I think that's really great, you know, and I think this is and we probably will, as a community, discuss where terms be, you know, revised to include certain things like podocytes in both definitions versus how to incorporate embryology into it, which is really the source, right? So I think this is really great. So thank you. I think this is exactly what we are hoping we can achieve. Yeah.
Chris, this is Todd. If you could comment on, this is a fantastic way for us to look at the relationships, the figure you've made here. But how difficult is it to generate that? This is a pretty small structure as an example, and it's a very good example. But as this grows in complexity, is it difficult to generate these types of outputs?
Yeah, let's see. It's kind of not so difficult for me to do it, but I've got a slightly odd kind of pipeline that I've developed that's not very well documented over the years for doing this. It's a bit experimental. So I guess the honest answer is right now it's not as easy as it could be, but it could be made easier if we had dedicated resources in this direction.
Yeah. So
go ahead, Sanjay, I'm sorry.
I was going to say that probably is a good point, Todd, that, you know, to have somewhere like trainees or dedicated support to go with this exercise, probably for all the organs, we would need to do that. So it's a significant effort, I guess, right? Yeah.
Mm-hmm. And also, Chris, I know that in the ontology community, you guys have developed a lot of, and I've heard this from Melissa, a lot of tools to do your work. And it could be that tool generation is part of what's needed in this area. Would you say that's true, or?
Definitely, yeah. We definitely need better tools here. Mm-hmm. I mean, and also just training in some of the tools that exist already as well.
Yeah.
Yeah, and there's a lot of fairly good command line tools now, but you know, for the biologist to really be able to work closely with the informatics people, we need the visualizations, and that's where the customization that Chris has made for himself is something we've all been begging for our different anatomical communities for a long time. So it'd be wonderful to try to explore what we could do in that regard.
And I'm going to add that there's also the challenge of there's only so many discrete things like this, like a renal corpuscle among all these organs. A lot of organs are more nebulous in how they're constructed. So this is a great example, but it won't necessarily play out in all the other organs.
Right.
Yeah.
So yeah.
Can I ask something about the relationship between the Uberon, the structures, and the cell ontology? So you seem to have veered quite, and I don't know whether this is a feature of cell ontology or your annotation and so on, but this seems quite, the cell names are relatively sort of structurally guided. So glomerular mesangial cell, I mean the glomerulus and mesangial is referring to the place, but is that cell like a fibroblast type stromal cell, or is it epithelial? You know, it's like, to me that isn't coming through in the names.
Yeah,
so sometimes it is, sometimes it isn't. But what are the thoughts on that front? Because this was something that we actually debated when we started working on kidney single cell RNA sequencing, you know, I think four or 5 years ago. And there was confusion because I look more at the basic, I'll say, characteristics of the cell, and the clinician that I was working with was looking, Mena Clatworthy.
Yeah.
You know, she was looking more at the where the cells are and so on. We kind of had misunderstandings and disagreements sometimes on that front.
So what you'll see a lot of time is what we call grouping classes. And you know, you're probably often more interested in classes that are under, you know, the leaf nodes that are underneath that are the more specific cell types. And in some cases, we just haven't really fleshed those out, and we need to do more work on the ontology and include those more specific types. I will also say just
okay, so you're saying the kind of coarse-grained classification.
Exactly. Yeah.
But there's like more detail within it.
Exactly. Yeah.
Okay. Fine.
And there may well be more detail here. I didn't draw the graph any further than the ones that were already in the ASCT table. And I'll also say just the kidney is, there's just the kidney cell types in the cell ontology they in particular have a lot more of these grouping classes just due to the historic nature of one particular project that was really needed those, and they basically needed a cell type for every anatomical structure. So there's a bit of redundancy, what looks like redundancy between the cell types and the anatomical structures, and you'll see a lot of things that are just like cell of kidney, which is to me that's so vague. It's not really particularly useful, and you know, we just probably at least want to kind of have some way of filtering some of these less useful grouping classes out. But yeah, the kidney in particular is in the cell ontology is a bit unusual for that feature.
Would you say that there?
Okay, I'm with you.
This is a great
Go ahead.
And actually, I would love to also thank again all those which created the initial tables, because we didn't give you much guidance. So going forward, I think there's a plan to give you whatever ontology terms already exist in Uberon and FMA, so that you can work from them, and then maybe naming differences won't be so dominant. Also, as you might see on this slide, Chris pointed out that cross-checking against existing ontologies could probably be automatized for all of these tables, meaning that whenever experts had a chance to review those tables, we can then also cross-check them against the ontologies. And then the second check that also needs to happen is the cross-check against the 3D reference organs, which some of us have been working on. Also in Spark, you have so-called scaffolds. And so I think these two checks can be done semi-automatically. And I think the construction of those tables can be guided by what knowledge already exists in the ontologies that have been developed by many, many experts over many, many years with a lot of lifetime going into them.
Yeah, and I think, Kati, what at least in, the kidney, what we're doing, and Todd is part of that, and I think Melissa maybe too, is going after each cell type or term, the definitions with you know, a lot of community present, to see which definitions are supposed to be newly added versus edited and revised, because there are also inconsistency in the existing database. So I think the only way to do it is really curate, you know, line by line, all of those things with, you know, it's a huge effort, but at least for the kidney, we are starting doing this now.
Yeah, and it's great to see the results. And I think what's interesting is that the AS, the anatomical structures, they are hierarchical in HubMap for means of browsing. Of course, on the back end, there's the entire graph which you can use. But for means of the exploration user interface, it is a nested tree. Also for the cell types with the cell ontology, we just had a really great call with Rahul from our team, who is leading the other mapping component in HubMap. The cell ontology is also hierarchical. So you can take all of the immune cells and you can select all of them by just selecting immune cells. So those two are trees, at least in the interface. But then the mapping between the two is a M to N mapping. And then, as some of you know, we are very interested in pursuing a vasculature-based common coordinate framework. And that vasculature network is a network. So we have two trees and two networks, and it all has to go together. And this is going to be a true user interface challenge. And I love it because we love building user interfaces that are easy to use. So I think it's really great to listen in, and I'm going to continue to listen in. And I think the next organ, just in the interest of time, is the liver. And I think Anna or Luke, who would like to present this one?
So what the approach we took was really starting from, since we were trying to link the cell type and the markers, so we are kind of granular level to the molecule, to the cell, to the tissue, and to the organ, we started to look at the functional unit of the liver. And so we started with the liver lobule, that is the first part. And then from there, we start to, dissect the liver lobule and trying to check which were all the other parts going from there to the cell level, and then linking to the marker that has been used to define those cells. And so here you will see all these additional parts. And I add the Uberon because I check whatever was already present in Uberon and in the cell ontology. And most of the anatomical parts are already in Uberon. And the same thing for the cell type. There are some differences, mainly because there are cells that are cell type that are very specific on where their location is. And so it's the case of, for example, periportal hepatocytes respect to the general hepatocytes. And so in this case, I think what I was going to do was kind of, this could be added as a children of the hepatocyte in the cell type ontology. And so there are other cases where mainly the cell type is different depending on which location is on. And that's, it makes sense because depending on which location specifically, if it's more close to the vessel, the artery or the vein, has different function in the organ. And so the function are reflected even by the cell type and marker that they express in that particular location. So that's was one of the things that we found. There are few cells that are not in the cell ontology, but just because they are very specific. But there is the parent cells, like the hepatocytes is there, in this case of the periportal. And then the other issue that we had was about how to represent the, no issue, was a kind of question. How should we representing the non-cellular component, like the stroma and the connective tissue? That we have one point in the table, yes. Yeah, sorry, but otherwise, yeah, it's down, and we thought it should be in put here even if it doesn't have any cellular component, but we'll have a specific marker to be recognized, and that will have a specific structure, and the component of the connective tissue are really important at the level of the function of the organ. So we add that too, as obviously we put only the molecule under the kind of marker, even if they are not cell marker. And so our question was, do we need to do it and add this non-cell structure? And if you think just, if we should kind of decide on another kind of column to put additionally to what we have in order to represent that. And this is a general, because I think that's, all three of us, we think is the important component to add. But then, you know, we can discuss that. And the other thing is, if you just go down, there will be all the immune cell here, yes, all the immune cell, but we only listed the immune cell that are resident, even if they are few, but still resident in the organ. We didn't put all the cells that are coming through the blood flow, because I think that will be common to most of the organs. So these listed here are only the resident immune cell types that we found that should be linked to the liver. And these are the specific markers. So that's why we put this as a side. And these are still, since I've been working with the cell ontology, now 11 years ago, I think I published the first paper on how to represent a cell and taking this dendritic cell ontology example. We are now, I talked with Alex Seal, that is the cell ontology person, and David maybe knows too, that I'm going to revise all the dendritic cell parts to update it with the new marker, a new cell type, if they are subtype of the dendritic cell. So all this part is going to be revised from the cell ontology side. And I know that they are going to revise mostly of the immune cell type in there. So that's why these are not still completely, do not have any cell type specificity label.
Thank you so very, very much. I think this is really interesting to understand how different organs are represented quite differently, and then different questions and issues come up. So if you would like, and if you have time, it would be great to have some of these design decisions, which you just explained, and also revisions that were made after you got feedback from other experts, captured here. What we have learned in HubMap is that we can oftentimes learn from one organ society and help other organ experts to do similar tricks, and also give some tips basically across these organ silos. It's also wonderful to see that you already started having PM IDs listed on the right-hand side. As you will see in a moment when we go over to the standard operating procedures for the table construction, what we would actually love to have is not only the body parts themselves, but we would love to understand what are the differences across the spectrum of individuals as we age, for different ethnicities. So I think this initial table is just the beginning of a much larger exploration into human diversity. And yes, we will want to know where it was published so that we have a track record of who found out about a certain type of piece of wisdom of truth. So in the interest of time, are there quick questions to the liver? I know we have a number of liver experts here.
This is Melissa. I just have a quick question, and we may not have time for it, but I just want to log it for later, and we could follow up if you like. So one of the issues that we have with the ontologies is the use of biomarkers in the definitions, because these ontologies are cross-species and cross-development. I often think that the strategies that we have for the cell ontology really should be thinking about different approaches where we have either, you know, age specific or context specific or taxon specific kind of, you know, annotations or rules or subgraphs that include those biomarkers. And the fact of the matter is that's exactly what's needed to reconcile the ontological framework with the sort of data-driven biomarker-based classification. So I think it's really a key area of sort of informatics research, and I was just wondering if you all had discussed that, or if you had some strategies in mind, or if we should just have that be a whole separate discussion.
I don't think this has been discussed yet, Melissa. I think it's a great topic for the next meeting number 3, and I think we can all together prepare that so that we can also present what's known on it. But it's a great question. And maybe somebody else on this call has other comments on it.
I could say, if I could just say briefly on this, so we already started to add, in some cases, organism-specific terms. We have ways to add organism-specific markers to the ontology, but I think for annotation purposes, it's useful in CL to have organism or species-specific terms as well in some cases. So there's already been some work that Alex Deal and others have been doing to divide up all of the markers that we have for immune cells to make clear what's human and what's mouse, because they were tangled before. So I think we do have strategies for adding those directly where they're really clear markers. In other cases where it's sort of clinical markers and there isn't a clear single gene mapping, I think we probably need other strategies.
I have a quick question. I remember there was even an app that allowed people to categorize immune cells. Is that still available based on biomarkers and types and things like that?
This is Bjorn's app.
Yeah, I'm not sure. I mean, several years ago, I know that it was already available. So is that somewhere available still?
I'm aware of a few different efforts. There was Ryan Brinkman's as well. Do you know, Anna?
No, I really, I don't know about the app, so that could be my ignorance. I don't know.
I was surprised. It's not my area of expertise, but I remember I went to some meeting and people say, well, you can download the app and any biomarker you have, you can immediately, or any type of cell you want to see what biomarkers correspond to that, you can. And it was only for immune cells. Anyway, I'll try to dig out.
What you're talking about is ImGen. That's mouse immunity, Serena.
Maybe,
you're talking about the ImGen app, and that is super useful actually. But it's focused on the mouse immune cell compartment, if you will.
Okay. Because I remember it was quite an advance. Yeah. Mm-hmm.
Yeah, ImGen, and they had a developer focused on the mobile phone app and so on. It's led by Christophe Benoit at Harvard, and the original focus was bulk RNA sequencing of purified mouse immune cell populations from different tissues, lymphoid as well as non-lymphoid, I think, if I remember correctly.
Yeah, and I even think with the monocells will be kind of more complicated unless you don't want to have the general type. But I think that the immunocells are very context specific, and that's why I like all these projects, because that could be a way where you could improve that part too. So you can have a general cell class for a cell type, but then there will be, like we have found some of the other cells that are very context specific, and that's due to the fact that the immune system, really the immune response is dependent on the context. And so that's why maybe we have something to think about, and this project could lead us to that too.
Very good. So we have 4 more tables that could be presented, but we also have other materials that we wanted to discuss today. From the table presenters, we have lymph nodes, we have heart, we have brain, and we have small and large intestine. I know the colon and the intestine should be presented today, but what other organs would really like to present today? So maybe I just ask the intestine team to present. I think it's John Hickey who is going to present. And then we will move all the other 3 tables to next time, because I think people have spent quite a bit of time and work on those tables. And I think it would be really great to hear what they learned and what went well and what was challenging. But in the interest of time, let's do 1 more table and then go over to other materials for today. So John Hickey, please.
Katie, thanks. So just to preface, Ying, who's not on the call, he has to be in a transplant surgery today, did a lot of work on this as well. And I am by no means an organ expert on the intestine or colon. A lot of my knowledge comes from actually doing codex multiplexed imaging on these tissues for the HubMap consortia. So while I was filling them in, I guess just some lessons that I learned. is that throughout the small bowel and colon, there are a lot of repeat structures that the small intestine colon gets subdivided into, like the mucosa, submucosa, and serosa, and muscularis propria. And a lot of these substructures are very homogeneous across the tissues. There are some small differences, which maybe I'm not as keen to, where further expert editing will be necessary. But a lot of these tissues have similar cell subtypes. And so copying and pasting, you see here, you know, I filled it out for one subregion of the small bowel, the duodenum superior. And so, you know, I guess my thoughts as an initial, you know, person trying to put something like this together where I haven't done this before is, you know, how could it be structured differently where we don't have to, you know, copy and paste all the similar structures across organs and potentially minimize that. So I don't know if you had something else for me to present, but that was just something that we had thought about while we were putting this together.
Yeah, so it would be great if you could capture some of the design decisions and also the challenges that you encountered on this particular organ, which might be one of the longer organs actually, if you could capture them in here. And again, I think what we are seeing is that these organs are very, very different. Experts have very different view of these organs. Some of them might look at it from a developmental perspective or from a disease perspective. What we need for HubMap is adult and non-diseased. And so I think these calls are also trying to get you all into the same mindset so that these tables are ultimately generated by experts that realize, okay, this is for adult and it's for non-diseased right now, but then of course with major applications for also understanding disease. And again, I'm very appreciative of the much work that has been done here in very little time. And let's see if anyone else had comments, questions on this particular table.
Yeah, I just wanted to make a comment. I agree with the general principle that there are repeating structures along the length of both the small intestine and the large intestine, but there are also regional differences, in particular in the epithelium. And I think it's important the table captures the regional differences because the epithelium fulfills rather different roles in different places along the gut.
Yeah, definitely. Katie and Mark, you know, in terms of what fashion could you just enable editing access by, you know, could you just edit that versus, because right now just Ying and I have edited it. Is this something that this big project is going towards, where we enable editing access for everyone? Because this is something, you know, it goes outside my area of expertise. So, I mean, I would encourage maybe you to edit it as you see fit. But yeah, I don't know what your thoughts are there.
Sure, I'd like to, I can do that. In fact, I sent an email a couple of days ago on this. I'm not sure if you've seen the email I sent, but I did send a number of comments a few days ago.
And if you copy the current table over to this link which you see here, then anyone should be able to edit it. And I realize that this is a very primitive, non-editorial controlled process right now. We are in the process of bringing structure to all of this. And this is actually a great lead over to the next agenda item for today. One is that if you have authored or reviewed one of those tables, please do make sure to add your information into this document so that we know more about you. We can also reach you. We can acknowledge your contributions. And then we also have started to create a document for a more structured editorial process to curate those ASCT tables. And again, I don't see this as a one-time effort. I think this is an effort which will take us years to do together. It's a complex organism we are trying to map. So if you go to that link, you will see an introduction into why we even want these tables and where they came from originally. there is information on the table format, and you might have realized that there are there's a left and a right to those tables. On the left are the anatomical structures, on the right are the cell types. On the more right are the assays that can be used to identify the cell types. So there's basically a left, a middle, and a right, and that's explained here in a little bit more detail. And then there is a last set of rows, and we are trying to get examples for this, which would actually explain the vasculature that is present in each one of those organs as well. And then the next section in this particular document explains the nested objects view, which we have for the anatomical structures for HubMap currently. And so you really have from the whole body to the organ, to the functional tissue unit, to substructures, to the cellular level, and a way to navigate down in a hierarchical way, and up again, of course, also. What we want to have in the future, and we are not there yet, is to have indications of how many, for instance, cells of a certain type, or how many glomeruli you actually have inside of a certain body part. So we know that there are about 1 million glomeruli in each kidney, left and right. So we are working on a better way to represent that. And again, I think we can learn massively from ontologies that already exist. We are also aware that other efforts have started to create interfaces so that people can upload the number of cells or the density of cells for different, here in this case, mouse brain regions. If you have seen of other efforts that try to get this kind of information by other means, please do paste it to the group chat window. We are going to harvest it from there, or send us an email. We have given you also an email address that you can use to reach us. And similarly, also, we would love to have information on the sources. And you still see my comments in here, how to best capture all this information and how to make it easy for not just one person, but many, potentially hundreds of experts, anatomists, pathologists, and others to help capture this information. And I think this is a great opportunity for really pulling together the community and having everybody also, of course, benefit from the results of this effort. So I think that is what these calls are ultimately about. As you know then, these ASCT tables, they are ultimately used to create ontologies, these so-called anatomical structure autonomies, and also the cell type ontologies. Again, these two are hierarchically organized, so they're trees, but then they are also matched against the 3D reference object library under development within HubMap. But of course, they're also matched against the cell types, also those which Rahul and his team are harmonizing across different labs and technologies and individuals. Ultimately, again, there will be also a matching between cell types and anatomical structures. And of course, there's also the vasculature, which interlinks those all as well. And we don't have a totally great solution for this yet, but I think we are working towards one. Then for the construction of the tables, we believe that ultimately there need to be experts. Initially, it was Jim G and Peter Hunter and myself, which have to invite leading experts, organ experts, to contribute to the design of the ASCT tables. And so far, it has been wonderful to work with all of you on those tables. Then we need to find a way to acknowledge the expertise that was provided. So there needs to be a true authorship. And then there needs to be a proper review process so that it's not just emails, but there's a system in place to rigorously basically collect all this information and then also submit it for review, get the review comments compiled and shared back with authors, and to do this for potentially very large author teams, because as you might have realized by now, it's rarely that one person knows the entire organ. There are bits and pieces that probably are distributed across multiple brains, and so we have to find a way, an infrastructure, and design an infrastructure to capture that information. Currently, some of it is in human heads, some of it is in publications, going back in time. But I think it's a massive undertaking, and it's very similar to the ontology development efforts that have been ongoing for many, many years. So we should join forces, and it's especially nice to see Chris and Melissa and also Mark Moosen on this call. And I hope Mark can say a few words when I'm done with this overview here. In terms of review by CCF experts, we thought a monthly cycle might work well. So we would review how many tables came into existence. We would then submit them to experts. So we are still looking out for more experts for the different organs. So if you're available, interested, please also put your name into the group chat or contact us later. There is this info ccf@indiana.edu email, which you can use to just volunteer your time. And again, thank you for that. And you will get to see maybe the very first ASCT table for the organ you deeply care about. So there is some benefit in volunteering to review. And then we would also ask authors to review or to revise their tables, and then to share back until there is a table that can be shared publicly. And then we are interested to work with Melissa and Chris to create views of these different tables. And then ultimately, again, these tables would be made available freely for anyone to use, ideally as subsets of Uberon or FMA or CL. And we still need to understand what that exactly means and what kind of subset this should be. Should it be an NIH-HCA subset, or should it be an ASCT subset? So what kind of naming to use there? And I think that all needs to be decided. But ultimately, it will be incredibly valuable to have those tables for HubMap and hopefully also for the other efforts. So I'm going to stop here and see if there are questions on this document as it exists so far.
I have one question about the representation of a different relation other than the ISA and NS-PARC. So if we look, we have to think about design which relation, looking at the both 2D and 3D organs, because I think they could be sometime different type of relation. If you see, like, you know, in a 2D a section of the slide of a liver respect to if you see in 3D. So we should kind of consider the both cases, because maybe we want to use it for pathology as well as for, I don't know, other computational uses in 3D. Or we have only to think in 3D? That's my question.
So many of these data sets come into existence because you have slices which then get re-rendered here, for instance, by Songling Ding from the Allen Institute into a 3D structure, and then each one of those 3D objects gets named. A similar process was also used by Kristen Brown, NIAID, in collaboration with our team to get us these kidney reference organs. And so I think over time we will have both. We will have the 2D representations and we will have the 3D representations, and we will have counts of the different structures, and we will have the Uberon corresponding or CL corresponding namings for these structures. But let's see if there are others which would like to chime in.
One point that I wanted to make, and maybe it's a small one, but it looks like we're talking a lot about the tissue ontology, like the organ ontology, and then the cell ontology. I know that in these tables that we're filling out, we're also adding markers. And so should we also be including the gene ontology or the protein ontology?
So I can try to take a whack at that. This is Melissa. So, you know, I think there's, and there's a lot of chatter in the text chat about the same problem. So I think you absolutely should be recording this information. I think what, you know, and David had mentioned this as well, there's multiple approaches to, so first of all, Uberon and CL are fully integrated logically. So cells are defined in part based upon where they are anatomically and vice versa. So that part is all fairly robust, even if it's not complete for any given portion of tissue. But what's harder is the fact that we've tried earlier in the CL to represent cell types with biomarkers, but the problem is those biomarkers tend to be either not representative of all the cells that are defined by that type, or at all stages of development for that cell type, or in all taxa, and there's no taxon representation. And so it ended up getting things quite confused. So that was not, you know, this is an older experiment in some ways. We need to sort of disentangle those definitions in a different way. And there's multiple things that can be done to ensure a robust classification according to the various biomarkers. As far as integration with the gene ontology, the gene ontology is also well integrated with in CL in terms of if you have a cellular or a tissue level function or a subcellular component. So that part's already readily built in and can be used very easily in the definitions. The problem really is the biomarkers, and I think what would be helpful to us in designing the most robust approach, which is really needed to leverage the full reasoning capabilities of the ontologies in combination with the data-driven cellular classification that you come from the single cell and slice data and these kinds of things, it is to say, is that presence of that biomarker or that protein or what have you definitional, or is it you know, an indicator that it may be one of those cell types? And that's the problem, is that oftentimes we can't tell the difference when we're designing these systems. So any information that you can include as a note, or just to get us started on what are the various attributes and how would that information be used in such a system, will help us design a collaborative approach to how to use that information. Does that make sense? This is one of the biggest challenges that we have right now in this field, in my opinion.
Sure.
Yeah, thanks, Melissa. I can add to that is that, you know, that biomarker question is going to be an iterative process. Like the table we did for kidney, we did from the data that we had at that time point of those biomarkers from 2-3 different technologies of single cells and more than one technology, you know, seeing those markers. But as the granularity is increasing, you know, and the platforms or the way you interrogate change, so this will be continuously be revised, you know, and how people analyze these things. So, and the truth, you know, the ground truth is going to be coming from the validations on the 2D, 3D, you know, multiplexed in situ or immuno or those kind of approaches to, you know, really show that if those, you know, annotations based on single cell data are true or not. But I think it's an iterative but a very challenging process. Yeah.
I would also like to just add that given the work that I've been doing with Kadi's group, looking at Uberon and cell ontology and thinking about the molecular biology and biomarkers, I think even if we could just get the very well established biomarkers properly annotated and put into cell ontology, that would be an amazing start. And it's not all perfect right now. So even just that, and then we have a good foundation to build on then. My 2 cents.
Yeah, I mean, we might be able to do it slightly outside the cell ontology and just have it as an imported thing so we can make a start quickly. Sorry.
No, I just want to go backwards kind of on the marker question. And basically, I think there are two kind of angles on this. One is, you know, cell states, cell types, and markers that are kind of freshly defined and discovered through the new methods that we have in single cells and spatial methods and so on. But then the second thing is that, you know, markers are traditionally really important for like traditional medical pathology and histology and so on. And what would be nice, and what we've been working on in England, is trying to link up those worlds somehow. So you know, what are the traditional immunohistochemistry, antibody kind of markers that have been used in the clinical community, and how can we link that, because that sort of speaks to the big medical community. I think Mark is it Halushka? I'm not exactly sure how to pronounce your last name, but I mean, could you have something to say about that?
Sure. Yeah, I completely agree with what you were saying, which is great. So I am a cardiovascular pathologist who signs out clinical cases, and there are a series of probably well over 150 immunohistochemical markers that we use to classify cell types, predominantly in cancer, but also just in general. And Sarah's right. The better we can correlate what comes out of the single cell experiences or defining of cell types that correlates with what pathologists use, the more this will be well received in the medical community and can be used for patient care and just the understanding of where things are. So I think that's a fantastic point that you're putting together.
Mark, this is Todd. If you could comment on whether or not some of those markers would have to do with something like collagen deposition, which is not cell specific. So one complexity I see is how you just or get that connected to a cell type when it could be coming from several cell types in an area of tissue.
Sure. So for something like extracellular matrix, I don't know how you would define that either, because you're right. In the matrix, it's a protein, and that protein came from a gene. You'd have to go back to the mRNA level and see where the mRNA was derived from. If it was a fibroblast or a smooth muscle cell or some other cell type that's present a parasite. So I really can't answer that in the extracellular matrix, but in general, for cell-specific expression patterns, there's a lot of usefulness, and there's a lot of questions that if we had better markers that came out of this type of work, they could be incorporated into better patient care.
But I do want to say that in pathology, it's used the quantity, organization, localization of extracellular matrix is used as a marker for tissue normality or abnormality. So even if you don't know exactly what are the cells producing it, although you can probably guess many times, the amount and the organization and the localization of extracellular matrix is normal or abnormal, and there are stains for that and so forth. So
is it reasonable to have a starting point of limiting some of the markers, even the classical ones, to markers that you can tie to a specific cell, at least that level of resolution, and then worry about how to add on ECM in general as we move forward?
But even for ECM, we know what are the major producers of ECM. So depending in which tissue you are, there are many, and coming back to what Sarah was saying, and I remember having that discussion also at the HCA meeting, coming back and being able to relate to what we already know. We already know a lot about all these tissues and cells and what they make and how you can identify them. So I think the new technologies probably will take us even further, but being able to confirm what we already know for so many years, I think that would be a great goal. So for collagen, depending on what type of collagen it is, if it's type 4, it's usually basement membrane. It could be an epithelial cells, endothelial cells, whatever. If it's type 1, it's usually a fibroblast or smooth muscle or whatever. So all these things are actually fairly well known. And they will be probably findable again in the profiles from the single cell analysis. So you could guess what are the source of these extracellular matrix components.
So I know we are up to 1 hour, and I know some of you might have to attend to other meetings. Thanks so much for coming in. We will share the recording of this session. I think there's also sufficient interest to have another session, so meeting number 3 in, let's say, 4 weeks. That would be June 4th, not July 4th, but June 4th. And I will just invite everybody again. Feel free to bring others that might be interested in this discussion. And I think we will just move some of the agenda items from today to a next meeting. But I think there are also a number of really neat new ideas that were brought up. And I will try to find organizers and kind of leaders for those discussions. Thank you all for coming in. It was great to see you all.
Thank you, Kathy, for putting it together.
Yes,
Kathy, thank you.
Thank you so much. Bye.
Thank you.
all right.
Thanks, everybody. Nice to see you.
Thank you. Bye-bye.
Nice seeing you all. Bye. Bye.
Bye.
Thank you very much, Katie. Thank you. Bye.
Yeah, thank you, Mark. Great comments.